Alright, welcome to another video. Okay, so basically Miss Nghi is going to present an IMAT topic, biology, or what is it gonna be? Biotechnology and some bioenergetic, right? Do you need time for that?
Yeah.
Alright, I'll give you some time to prepare for that part.
Yeah.
Okay, let's do it now. So Miss Nghi, please.
Now I'm going to talk about biotechnology, which is about DNA recombinant. DNA recombinant technology involves using enzymes and various laboratory techniques to manipulate and isolate DNA segments of interest.
So why do we need to cut and then stick DNA fragments, and why do we need to manipulate that?
Because we want a specific DNA and we want to introduce it into other host. Or we can introduce new traits into new organisms or create new genetic combinations out of DNA recombinant.
Okay.
So there are 7 steps of DNA recombinant. First, we need to isolate genetic material. Then we insert the restriction enzyme. Then we use PCR for amplification. Step number 4 is ligation of DNA molecules. Number 5 is inserting the recombinant DNA into a host. And number 6 is isolation of recombinant cells. So first, the DNA template is heated to a high temperature, which is around 19.5 Celsius, to separate the 2 strands out of the double helix. This process makes the individual strands accessible for the primers to bind. Then, short pieces of DNA called primers, which are complementary to specific regions of the template DNA, are added to the mixture, and the temperature now is lowered to between 50 Celsius and 65 Celsius in order to allow the primers to bind to the target sequences. So the primers serve as the starting points for the new DNA strand to be synthesized. Then an enzyme called DNA polymerase is added to the mixture, along with free nucleotides, serve as the building blocks for new DNA strands. After that, the enzyme synthesizes new DNA strands using the template, resulting in the reproduction of 2 copies of the target DNA sequence. And then the process is repeated multiple times, leading to the exponential amplification of the target DNA sequence. Okay, so that's it. So the application of DNA recombinant is in medical field. It helps to produce vaccines and protein therapies such as human insulin, interferon, and human growth hormones.
Okay, let's talk about insulin. Let's talk about insulin for a while. Why is there a need to manipulate the bacteria E. coli in order to make insulin for us? What is the need? Why is there a need? Tại sao lại cần phải thao túng cái con vi khuẩn E. coli để nó bắt nó tạo insulin cho con người? Why?
Because we cannot produce insulin.
Of course we can, but it will require so many cows, pancreas, and that is very expensive.
Yeah.
Nó cắt cái tuyến tụy của nhiều con bò ra để có thể có được cái lượng insulin cần thiết. Nó rất là mắc tiền luôn á.
Okay.
That is the reason. Okay, so next one, please continue.
Okay, next I'm going to talk about bioenergetics. First, I'm going to talk about ATP. ATP stands for adenosine triphosphate, which it includes adenosine and triphosphate group. And it is an essential molecule served as the energy currency of the cell. It powers various processes within the cell. So why ATP is the currency of energy? First, because it is a small molecule, so it can be easily transported within the cell, allowing it to be used wherever it needed. Second, ATP is highly reactive, so that it can readily
reactive.
Meaning that it can readily release its energy when needed. And ATP reactivity allows it to release energy quickly and efficiently, making it a suitable source of energy for rapid processes within the cell. Third, ATP can be synthesized and broken down quickly, allowing the cell to quickly replenish its supply of ATP as needed.
Okay.
So now I'm going to talk about how it can release energy. So the energy stored in ATP is released through the process called hydrolysis, which involves the addition of a water molecule to the ATP molecule, and it results in breaking one of the phosphate bonds, and then it releases energy in the process, which at the end it can create ADP, ATP and energy, a proton and a phosphate group connect with OH.
Okay, so where are these components going to?
Pardon? What component? Yeah. Sorry, can you repeat the question?
What I mean is those components that you just mentioned. What happens to them? Điều gì diễn ra với những cái thành phần mà em vừa mới nói đến? Nó đi đâu chẳng hạn?
So from ATP, when we need to store energy, we can turn it into ATP.
Okay, continue, please.
Okay. So now I'm going to talk about redox reaction in living organism. So redox reaction stands for reduction oxidation reaction, which are reactions that the oxidation state of molecules change. So the substance that lost electron is called the reducing agent. The substance that gained electron is called oxidizing agent. And oxidation is the loss of electrons, and reduction is the gain of electrons. So why is redox?
You said oxidation is the what?
The loss of electrons.
Okay, oxidation naturally means like gaining oxygen, which means you are gaining a negative component, right? So did you say something about the loss of electrons?
Yeah.
Coi kỹ lại đi, có phải là oxidation là the loss of electron không?
Yeah, phải.
Check. Oxidation. What does it mean? When a reactant loses electron during a reaction is called oxidation. Yes. Mất đi một electron thì cái số oxy hóa nó tăng lên. Đúng rồi, yes, exactly. So when oxidation number increases because of loss of electron, that is oxidation. Very good. Good. Yeah. Okay. And how about reduction?
Reduction is the gain of electrons.
Right, so it means that it decreases the oxidation number. Nó giảm số oxy hóa xuống. Very good. Okay, please continue.
So why is redox reaction is important in living organism? First, it produces energy and it helps us to metabolism.
No, to metabolize. The verb is metabolize. Động từ là
metabolize. To metabolize. It also helps to detox within the cells.
Detoxify, yes.
Detoxify within the cells.
Okay, continue please.
For example, redox reaction that produce energy, you can see in cellular respiration, where glucose is oxidized to release energy in the form of ATP. And during this process, oxygen acts as the oxidizing agent. And yeah, that's it.
Right, next part.
Can I have some time to prepare?
Okay, so what's the next part about then?
Photosynthesis.
Of course, it's a huge part. Okay,
yeah.
So did you talk about Krebs cycle and electron transport chain?
No, I didn't.
Đường phân đó em. Glycolysis, Krebs cycle, and electron transport chain. These three things are the main thing in the pathway of breaking down sugar. You know, you have to mention that. So when you mention about photosynthesis, you have to mention that where it happens in plant cells. Và nó chia ra, it has to be divided into like light dependent and light independent reaction. Phản ứng phụ thuộc vào ánh sáng, phản ứng không phụ thuộc ánh sáng. Pha sáng và tối á. And then inside of them you have small little details in there. Okay.
Okay.
Right. Okay, you have some time now. Okay, time's up, please. Present yourself about bioenergetics again. I mean, other parts that you haven't mentioned.
Okay, now I'm going to talk about photosynthesis, which is the process by green plants, algae, or some bacteria when they convert light energy into chemical energy in the form of glucose. So photosynthesis have two stages, light-dependent reactions and light-independent reaction or Calvin cycle.
Very good.
I'm going to talk about light-dependent reaction. These reactions occur in the thylakoid membranes of the chloroplast of the plant cells, which they involves the absorption of light energy by chlorophyll, and by that they produce ATP and NADPH and oxygen as byproduct. So this happens when light is absorbed. The energy used to transfer electrons from water molecules to NADP plus is a process called photolysis, and then those electrons used to power the synthesis of ATP and NADPH. So these reactions are also known as the electron transport chain. So yeah, now so at the end of now the product ATP and NADPH is used for the light-independent reaction or the Calvin cycle. So the Calvin cycle occurs in the stroma of the chloroplast of plant cells. It is the conversion of carbon dioxide into glucose
carbon
dioxide into glucose using ATP and NADPH So the light-independent reactions initiated when carbon dioxide enters the stroma of the chloroplast. So the stroma are often found at the back of the leaf. So during the Calvin cycle, the carbon dioxide combined with a 5-carbon compound called ribose biphosphate to form a 6-carbon compound called
compound
called glyceroldehyde 3-phosphate or G3P. So this reaction requires energy provided by ATP and NADPH, which produced during the light-dependent reaction. Then the GP3 produced during the Calvin cycle then converted into the glucose or other organic compounds through
organic compounds
through a series of additional chemical reactions.
What additional chemical reactions? My question for you is where does the plant cells get the ATP from?
It gets the ATP from the light-dependent reaction.
Right, but also in photosynthesis, there is also a there's also an electron transport chain, to produce ATP too. Cái quang hợp nó cũng có cái chuỗi truyền electron tự do của nó riêng nó nha để tạo ATP nha.
Yeah.
So that's why photosynthesis there's also ATP produced to feed into the photosynthesis, because it has its own electron transport chain, isn't it?
Yeah.
So what happens to the other thing? Like, is there an involvement of electron transfer molecules, proton transfer molecules like NAD or FAD something? Is there an involvement of these molecules?
No. Right?
All right, continue, please.
Okay. So, so the Calvin cycle complete 3 turns to produce 1 net GP3 molecule because 3 CO2 molecules are fixed to generate 6 GP3 molecules, but 1 GP3 molecule is used for glucose synthesis. So other 5 GP3 molecules are used to regenerate 3 RUBP molecules.
Rubisco?
Yeah.
Right, okay. You need to mention the name Rubisco as well,
Yeah, I mentioned
cái phần tử Rubisco nó rất quan trọng đó, ở trong cái quá trình Calvin cycle đó.
Okay. So we can see that the light-independent reactions and light-dependent reactions are interconnected through the transfer of energy in the form of ATP and NADPH, the flow of electrons, and the cycling of reactants and products. That's it.
Is that all? Seriously?
Yeah.
Okay, other part, and how about other parts that you haven't mentioned? Is there any other part you haven't mentioned? There's a part down below, I couldn't see it. Fermentation, yeah, fermentation. And
No.
anaerobic respiration. So did you mention about a Krebs cycle and electron transport chain and then glycolysis? Did you?
No, I think I will do it let me prepare, then I will do it.
What do you mean?
You
need more time or what?
Yeah, I need more time.
Okay. Alright, time to talk about it now.
Okay.
Right.
Now I'm going to talk about glycolysis. So glycolysis is a series of reactions that extract energy from glucose by splitting it into 2,3 carbon molecules called pyruvates. This process takes place in cytosol of the cell. There are two main phases, the energy requiring phase and the energy releasing phase. First, I'm going to talk about the energy requiring phase. 2 ATP are spent to form an unstable sugar with 2 phosphate groups, which then splits to form 2,3 carbon molecules that are isomers of each other. So there are 5 steps of this phase. First step is that a phosphate group is transferred from ATP to glucose, making glucose 6-phosphate. And glucose 6-phosphate is more reactive than glucose, and it traps the glucose inside the cell. Step 2, glucose 6-phosphate is converted into its isomer, fructose 6-phosphate. Step 3, a phosphate group is transferred from ATP to fructose 6-phosphate, producing fructose 1,6-biphosphate. This step is catalyzed by the enzyme phosphofructokinase. Which can
Phosphofructose kinase.
Phosphofructokinase, which can be regulated to speed up or slow down the glycolysis pathway. Step 4 is fructose 1,6-biphosphate splits to 2,3 carbon sugars, dihydroxyacetone phosphate, which is DHAP, and glyceraldehyde triphosphate.
Glyceraldehyde triphosphate. So basically, after 5 steps, it uses up 2 ATP for that process, right?
Yeah.
Nó xài 2 ATP ở qua năm bước đầu tiên, xong rồi năm bước cuối cùng là được nhận 4 cái. So after the first 5 steps, 2 ATP are used up, and the last 5 steps, 4 ATP is being produced. So generally, you have 2 ATP being made after 10 steps of glycolysis.
Yeah.
Please continue.
Okay. So now we talk about the energy releasing phase. So step 6 is there are 2 reaction occurs simultaneously. First is the G3P oxidation, and second one is NAD plus reduced to NADH and H plus. So the overall reaction is exergonic, releasing energy that is used to phosphorylate the molecule, forming 1,3-biphosphoglycerate. Step 7 is 1,3-biphosphoglycerate donates one of its phosphate group to ADP, making a molecule of ATP and turning into 3-phosphoglycerate. Step 8, 3-phosphoglycerate is converted into its isomer, which is 2-phosphoglycerate. Step 9, 2-phosphoglycerate loses a molecule of water, becoming phosphoenopyruvate, or PEP. PEP is an unstable molecule, so it loses its phosphate group in the final step of glycolysis. Step 10, PEP donates its phosphate group to ADP, making a second molecule of ATP. And as it loses its phosphate, PEP converted to pyruvate, the end product of glycolysis. Yeah.
Okay, that is All right. So afterwards, what's the result of glycolysis? And does it require oxygen for that process?
No.
That doesn't require oxygen. So where does glycolysis happen?
It happens in the cytosol of the cell.
Cytosol or cytoplasm of the cell. Very good. So what happened for two molecules of pyruvate? If in the presence of oxygen, this pyruvate will be converted to acetyl coenzyme A or acetyl CoA, yeah? And then it will go into the inside of Krebs of the mitochondria, so Krebs cycle can happen. Can you continue from there?
Can I prepare a little bit about Krebs cycle?
Of course, but we have only 5 minutes left now.
I can talk about it like surface.
Okay, sure.
Okay. So about the Krebs cycle, where so the products of Krebs cycle will be Okay. When the cell have access to oxygen, the pyruvate will convert to acetyl coenzyme A, and then it enter the Krebs cycle. This process is known as the aerobic respiration and is the most efficient way of the cells to produce ATP. So for the Krebs cycle, it will produce from one glucose, it produces 2 pyruvate molecules. So from 2 pyruvate molecules, it will produce 6 NADH molecules, 2 FADH2 molecules, 2 ATP, and 4 CO2 molecules.
Good. And that cycle is a cycle. So basically, a lot of molecules from your other sources can join in the Krebs cycle. Rất nhiều những cái phân tử khác từ những cái nguồn gốc khác có thể tham gia vào cái Krebs cycle nha. Like for example, fatty acids can actually join in that Krebs cycle, you know. Ví dụ như phân tử mà chất béo mà nó bị tách ra, nó có thể join vô, giống như kiểu mà nó hỗn hợp vô, nó join vô một cái lý do nào đó, nó có thể join vô được để tạo năng lượng á. Tức là từ những nguồn khác chứ không phải là từ cái nguồn mà từ cái đường một cái glucose đó nha. Cho nên cái Krebs cycle đó nó vi diệu lắm luôn á. Nó có thể là nó trộn, nó dùng cái nguồn nguyên liệu từ những cái thành phần khác, từ những nguồn khác luôn á. So Krebs cycle can actually use the material from other sources, not just that one glucose you start with, you know. All right, so the electrons so the electrons after these cycles are carried by NAD, right? NAD and FAD, fat, right? To where?
Yeah.
To the space between the inner and the outer membrane of the mitochondria. Nó đưa cái lượng electron nó đến cái chỗ mà nào? Đến cái chỗ mà giữa hai cái màng trong và bên ngoài của tư thể á. Màng trong màng ngoài tư thể. Và có chuyện gì xảy ra không? And what happens afterwards? It's electron-transport chain. Và cái electron-transport chain nó làm gì? So what happened to the electron-transport chain? It involves something called ATP synthase. Nó có một cái enzyme gọi là một cái protein, nó gọi là ATP synthase. So when ATP synthase open, nó trước khi mà nó mở ra thì là bên ngoài màng tế bào nó phải có sự tụ tập của H cộng đã. So before ATP synthase can be open, the outer membrane of the mitochondria, there must be an accumulation of H plus ion. So when ATP synthase open, H plus ion flood into the cell. Mở ra một phát thì là coi như H cộng đi vô, nó đi vô cái ti thể. H plus get into the mitochondria and it drives the production of the ATP from ADP and phosphate group, right? Nó sẽ lấy cái sự mà tạo ra ATP từ cái ADP và nhóm phosphate tự do. So that is electron transport chain, you know. And electron transport chain can produces a lot of ATP, up to 30 to 34 ATP, you know. So totally for the whole 3 cycle, glycolysis, Krebs cycle, and electron transport chain, maximum you can get 38. Some books they say 40. Some books they say 36. Some books they say 38. But I think 38 is the right number. Okay?
Oh, okay.
Alright, good. That is about it. So next part we'll talk about anatomy and physiology of humans and animals. Okay, you need to look at those videos in IMAT about that. Okay, có ít nhất 11 cái hệ luôn á. It's about 11 different systems: animal tissue and homeostasis, and then system. Okay, just watch my video. It should be enough. And if you are wondering about something, just search online for more. Okay. Okay, we have to go now. Okay, that's the end of my videos. Please like, share this video with your friends, subscribe to my YouTube channel, and press on the bell button next to the subscribe button so you can get notified about my new uploads, okay? You can also press follow my Facebook, but please don't add me on Facebook as friends, okay? Cảm ơn các bạn đã theo dõi nha. Các bạn hãy nhấn vào like, chia sẻ video này bạn bè các bạn. Nhấn vào nút đăng ký kênh YouTube mình và nút cái chuông kế bên cạnh cái đăng ký kênh đó, để mà các bạn sẽ được thông báo về những video mới nha. Các bạn cũng có thể nhấn vào nút theo dõi Facebook mình, nhưng nhớ đừng kết bạn Facebook mình nha. Thank you and see you. Bye.